Hi, today we are going to study about demonstration of sex chromatin. So let us start with the introduction. Sex chromatin is also known as the bar body. The bar bodies are named after the discoverer Murray Bar. In the Murray Bar, the name of the scientist, the bar name is given to bar bodies, and that bar bodies is these days called as a sex chromatin. This sex chromatin is formed during embryonic life. Like if you see the fertilized form of the sperm and the egg is a zygote, which will further develop into the embryo through the 2 cell stage, 4 cell stage, 8 cell stage, then mass of cells, blastocyst. So within 4 days of the zygote formation, the blastocysts are formed, and then we can see the appearance of sex chromatin that occurs at the stage of 4 to 8 cell. When the zygote divide from 4 to 8 cell stage, then the sex chromatin started to appear in the cell. Sex chromatin, here we can see that it will appear when there is an inactivation of one of the X chromosomes. If there is 2 X chromosomes, so one of the X chromosomes, when it is inactivated, then it will be observed, it will be detected. So this is the sex chromatin. Now identification. How can we identify the sex chromatin in the cell? So this is the 2 cells we can see in the image. One is epithelial cell, another is a neutrophil. So epithelial cells, if you see the nucleus, very clearly we can see in the periphery of the nucleus one bodies we can see that is called bar bodies. The dark color stain is the bar bodies. In the neutrophil also we can see neutrophil has a segmented nucleus, and in the segmented nucleus of the neutrophil the drumstick like appearance will be seen, that is also the bar body. So any cell which has a nucleus where we will be able to detect the sex chromatin through the staining procedure. They appear dark stained in the nucleus. Now how can we demonstrate the bar bodies? What are the samples it is required? So the samples required is a scraping from buccal mucosa, scraping from vaginal wall, and blood sample can be taken, where in the blood sample white blood cells are mainly preferred because other cells have a no nucleus. RBC has a no nucleus, platelets has a no nucleus. So white blood cells, which is one of the largest blood cells, is preferred where nucleus are clearly visible. Identification of bar body. Okay, so not identification, indication, sorry, it's indication for bar body test. Why this test is done in this patient? So because this bar body test is one of the cheapest test, very economic, and this is not confirmatory test. Actually it is a screening test which determine ambiguous genitalia, means where the sex characters are not clearly visible or not defined. So in case of ambiguous genitalia, this is the test which is requested in the lab. This test usually suggests when there is a delayed onset of puberty. Puberty starts after a different age in the male and female. So after 9 years in female, after 12 years in the male, puberty starts. So diagnosis of this delayed onset of puberty, it can be screened through this bar body test. Some disorder like Turner syndrome, Klinefelter syndrome can be diagnosed easily through this screening test. Now pathophysiology. So again you can see the image. One of the cell is shown in this picture, and the cell shows nucleus, where in the nucleus we can see the dark color body, that is a bar body, and if we can see under the microscope, so you can see it is a positive bar body test, and this is only visible if the sample is from female. So bar body is the inactive X chromosomes. Female has a 2 X chromosome. One of the X chromosome is inactive and can be demonstrated in the female somatic cells. Any somatic cells of the female can be taken as a sample, and where you can be able to demonstrate this bar body. The chromatin in the chromosome is highly condensed, and it can be observed as a dark staining spot at the periphery of the nucleus. So this is actually the epithelial cells, where you can see the dark staining spot nearby the periphery of the nucleus. So this test is useful or helpful to determine the infant's sex, where the physical features are not clear. Physically it is not visible that what is the sex of that infant. So this test is helpful. Presence of one bar body or condensed chromatin is considered positive. So single bar body or single condensed chromatin is considered as a positive. Now see the mechanism of appearance of the bar body. In the picture we can see the mother and father. From mother we get X ova, and the father we get Y type of sperm. So fusion of that, we got a zygote containing both X and Y type of chromosome. So it happens, the zygote happens. Now actually the male is XY and the female is XX, and XY male, their body, their cell do not show the bar body, and the female who has a 2X, usually one of the chromosome is inactivated, and we can see that the inactivated form of the chromosome in the female is visible as a dark spot, and we can be able to detect that that sample belongs to the female, or that the gender of the female patient is defined as a female. So any sample showing the bar body is defined as a female sample. So we see this one of the X chromosome has been inactivated, and this occurs during embryonic life stage only, approximately 7 to 14 days after the fertilization. But embryonic life, we already saw in the previous slide that 4 to 8 cell stage also you can be able to detect it. Chromosome XX in a female, one X is inactive, and sometimes the inactivated X chromosome is donated from the father also. Female usually has an inactive chromosome, and hence the cell inactivated X chromosome gives the offspring with the same structure. So this is the image. While male has XY, and so no bar body and condensed considered, and that's why we consider is negative. So this is the sample belong to the male sample, because there is no bar body. So male no bar bodies are seen, and female bar bodies is positive. Interpretation: this bar body test is advised for the screening of ambiguous or doubtful sex character, Turner syndrome female, who has a XO, means there is only single X, will have no bar body. Male with the Klinefelter syndrome will have a bar body, and female with triple X chromosome will have a 2 bar bodies. So female cell with 4 X chromosome has a 3 bar bodies. So if we need it, the results should be confirmed by chromosomal karyotyping. These kind of things, if the bar bodies are visible under the microscope, so we need to confirm with the chromosomal karyotyping technique. Thank you for the listening.